Now that you've split your cells, we're going to count them. So this is going to be a little hemocytometer lesson. Normally, just know that you would be doing the counting before plating, but because of the nature of the class that we have, we need the hood space for everyone to go through quickly. So you will be counting cells after you've actually completed your split to determine the number of cells that are available. And you're going to do this, you're probably going to have somewhere around 250 to 500 microliters of cells. And to that, you're going to add something called trypan blue. Trypan blue is an exclusion dye, which means if the cell is not viable, the dye can actually get into the cell and stain it blue. So if you have a dead cell, it's going to appear blue or bluer than a live healthy cell would appear. So to that volume of cells, you're going to take and add about 50 microliters of trypan blue, and you're just going to let that set for a minute before you actually load your hemocytometer. And so you're just going to pipette that directly into the cell mixture and give it a good little thwop with your finger in order to mix it well. Once it's in with the cells, the trypan blue is going to stain any dead cells a dark blue that will be easily visible. Just know that these NIH cells, especially with how we propagate them pretty regularly, they actually have a 99% viability rate, and those calculations are in a separate lecture that will be covered outside of this little demo here, but the trypan blue is going to be added, and then you're going to load your hemocytometer for cell counting. And so you're going to take your 50 microliters, you're going to pipette it from the trypan blue bottle into your cell mixture, and just know the trypan blue is kind of nasty stuff, so make sure you're putting the waste into the biohazard, and that if you spill it onto the pipette or anything like that, make sure that you're using some alcohol to get it off of there. So that the next person who uses a pipette, or if somebody grabs it that doesn't have gloves on, they're not getting trypan blue all over themselves. Then of course, always tighten down the lid as soon as you're done so that it doesn't spill accidentally. This is the container that holds the hemocytometer set. We actually have quite a few of these. There is a special slide which has got the two silver sides to it, and a special weighted hemocytometer cover slip. We do have a tendency to break these a lot. If you break one, just let me know. We have a stash of them, but it is not just some plain old cover slip. It is a special hemocytometer cover slip that has a certain weight to it so that you have a certain volume of cells underneath the cover slip on the hemocytometer slide. It's actually really easy to load one of these. Once you've done it once, it'll be easier to do it the next time. You are going to take about 10 microliters of the cell volume that has been stained per side on the slide. So that means you're going to need about 20 microliters total. So you're going to take and load each side with about 10 microliters. You'll have a lot less trouble doing this than my demo here, and this is like an example if somebody got some dye on the pipetter there and didn't clean it off. But you're going to be able to put the hemocytometer on the bench. I'm trying to show you how I'm loading it, so I'm holding it up very awkwardly so that you can see. You kind of slide the pipette tip under the cover slip into that little V area on the hemocytometer side, and then you just inject the liquid, and it goes underneath the slide, and you can kind of see when it starts to overfill a little bit, and then I just kind of stop pushing more liquid under there and pull out the excess. And so it's put a very defined amount of volume. And now I'm going to load. I usually load both sides, and I usually do count both sides. In general, you want to count somewhere around 100 to 200 cells. And so, if you need to dilute in order to fit into that range, great. If not, most of the time we do straight counts, and we usually are somewhere in there. And so now I'm just loading the other side. And then you're going to go put this under a microscope, and you're going to actually count the number of cells dead and alive that are there and do some viability calculations as well. But that's the loaded hemocytometer. Again, it'll be easier for you because you'll be able to leave it setting on the bench while you're loading it. Under the scope here, this is what you're going to actually see. And so what I count, based on the calculations that I'm going to describe to you, we are going to follow the 16 black, which is on the four corners. So it's on the upper right of your screen, and not the 25 that are in the middle. That didn't have any cells on it. But as you complete your cell counts, you're going to go ahead and take this over to the biohaz waste. You're going to spray down the hemocytometer and the cover slip, and then you're going to gently dry them with a chem wipe after you spray them down with 70%, and return them to the original little box. And then I'm going to show you where that little box is. And we have actually a number of sets, so however many people we have in the hood, we're going to have probably twice that out in the lab space, and so everybody would be able to have their own hemocytometer to do cell counts when they're ready. And then you just put it back in there, close the lid, place the trypan blue. All of this is found together in the cell counting supply drawer, which is located over on the right behind the benches. This is a little cell counter. So I usually do live cells on the left and dead cells on the right, and that way I can just count and click those as I'm looking on the slide, and then I zero it out before I return it back to the drawer. And again, the drawer can be found right behind the hoods, and it's labeled cell counting supplies right under that scope. And you'll see within this drawer there's a pipette rack, a tube rack, some pipettes, some tips, a bunch of hemocytometers. The trypan blue is located in this drawer, and the little cell counter tabulator thing is also found in there. And just zero it out before you put it back in there, or the next person can do it. But that's pretty much it. Any questions, you know how to find me.